And first target for antibiotics, obviously, cell wall. Okay? So we're going to group by the targets. First, perhaps the largest group of antibiotics that we know so far are beta-lactams. Before we move any further, I know that this can be overwhelming. Okay? This is why I make very clear what I want you to know. I'm going to specifically mention, I'm going to say, this is the class of antibiotics. Does that make sense? This is the class. So you need to know this class of antibiotics, how it works. Do you need to know individual drugs in the class? No, you don't. Am I clear? So among beta-lactams, there are 4 classes. Penicillins, cephalosporins, monobactams, and carbapenems. Am I clear? These 4 classes all belong to beta-lactam antibiotics. All these 4 classes have the same mechanism of action. Am I clear? How do they work? So, remember cell wall, bacterial cell wall. So you have glycan chains, remember glycan chains. Remember them? That have to be connected to each other by the peptide linkages. Does that make sense? So you take glycan chains, and using amino acids you form peptide linkages, you get proper cell wall. This is not a chemical reaction, it's more of a process. Are you following me here? So this formation of peptide linkages is facilitated by the enzyme called penicillin binding protein, or transpeptidase. Are you following me so far? We good? So, beta-lactams, all of them, contain a structural element known as beta-lactam ring. This beta-lactam ring will inhibit transpeptidase. Now follow this logic. If beta-lactam inhibits transpeptidase, does the cell wall form properly? No, it doesn't. There are no links, right? So cell wall becomes structurally weak and the cell pops open. Are we clear? Make sense? You can ask, okay, what's the difference between all these 4 classes then? Well, imagine the beta-lactam ring, and you have all kinds of chemical bells and whistles that sit on that, attached to that beta-lactam ring, okay? So these chemical modifications in those antibiotics, they determine whether they're broad spectrum or narrow spectrum. They determine the pattern of resistance, they determine the bioavailability. Does that make sense? So for instance, you take first generation cephalosporins, they're pretty narrow spectrum, mostly gram-positive. Third generation cephalosporins, broad spectrum, really safe, great antibiotics. That makes sense? Monobactams, very specific to gram-negatives. Carbapenems, broad spectrum, all act through the same mechanism. But these chemical differences, besides beta-lactam ring, determine they spectrum of action, blah, 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 all that kind of stuff. Are we clear? Make sense? Now, one interesting mechanism of resistance that I will introduce you to right now is this. Some microorganisms develop an enzyme called beta-lactamase, which destroys beta-lactam ring. Okay? That makes sense? Chemically modifies the drug. Now follow my logic here. If beta-lactamase basically changes the structure of beta-lactam here, can it inhibit transpeptidase now? No, because look, beta-lactam inhibits it. Once it's chemically modified, it's not a beta-lactam yet, already. Does that make sense? You see what I'm trying to say? Horse can walk. If you chop off the legs, it can't walk. Okay, so basically it chops off the legs of the beta-lactam. So what can we do about that beta-lactamase resistance? And this is super elegant. This is an enzyme. Can we inhibit an enzyme? Dude, of course we can. We were talking about competitive and allosteric inhibition. So what pharmaceutical companies came up with? There is a drug called clavulanate or clavulonic acid, which in turn inhibits beta-lactamase. So now look how it works. It's so brilliant. Look, when you have a microorganism that produces beta-lactamase, you add to your formulation clavulanate. So clavulanate inhibits beta-lactamase. Beta-lactamase leaves beta-lactams untouched, and the microorganism retains sensitivity to the drug, to penicillin or any other beta-lactam. And these enzymes are called, if it modifies penicillin, it's penicillinase. If it modifies cephalosporin, cephalosporinase. Carbapenemase. Does that make sense? Have you heard about the drug called Augmentin? It's amoxicillin plus clavulanate. Clavulanate augments amoxicillin. Clear? Good? Okay. Second kind of, it's actually going to be 1, 2, 3, 4. So these are 4 classes, right? So 5th class. Oh, it's going to sound so familiar. Glycopeptides. One example, vancomycin. Okay? So glycopeptides inhibit glycan chain and peptide link formation. That make sense? So remember, again, if we look at the cell wall, glycan chains are rows of bricks, and peptide links is mortar. That makes sense? So beta-lactams don't let the bricks to be attached to each other by mortar, so they basically remove the mortar part. Okay? Glycopeptides break the brick chains and remove the mortar, bless you. And finally, the 6th class here, the single antibiotic, which is a representative bacitracin. It inhibits the transport of building blocks. So now look what happens. Again, remember brick and mortar? Bacitracin stops the trox like the trucks that carry bricks and carry mortar. Am I clear? Does that make sense to you? Okay. Now, question. We figured that bacterial cells have cell walls. Anyone remembers human cells? Do they have cell walls? They do not. They only have membranes, right? So, next question. Kind of 1, 2 out of 10. If human cells don't have cell walls, while, well, eukaryotic cells, well, okay, human cells don't have cell walls, while bacterial cells do. These antibiotics, are they generally safe for humans? Yes. This is called high specific toxicity. Okay? So we can say that specific toxicity is high. Do you understand what specific toxicity means? These drugs are toxic to bacteria, but not to humans. Does that make sense? Good? Any questions on this one? Make sense? All right.